Okay, good afternoon, everyone. It just hit 12 UK time on my clock, and it's 7 p.m. in Thailand, where our presenter, Professor Olivo, currently is. Thank you for joining us, to all of you, and welcome to the 6th edition of the GSU Malaria Journal Club monthly series. So we organize these every month, as you know. Today's webinar is parasite focused, referencing Professor Olivo's recent preprint titled "A Complex Plasmodium Falciparum Cryptotype Circulating at Low Frequency Across the African Continent." For those of you who don't know me, my name is Pablo Chauke, and I'm a training coordinator at Malaria Gen, working with a team of awesome people to organize these monthly events. People that include Shri, Christina, Vicky, John, Alistair, and Chris, and myself as well. So thank you so much for joining us. Just a few ground rules about the Journal Club. The Journal Club is being recorded so that you are aware, and we will share the recording with the rest of you on our Malaria Gen YouTube channel later. So please do be on the lookout for that. Please kindly stay on mute. We will have a Q&A session at the end where you can raise your hand or type your question or comments in the chat or Q&A box. So during the presentation, please do pop in the questions that you are thinking about in the chat or Q&A box. That would be helpful for us at the end when we have the Q&A session. Without further ado, I would like to introduce our speaker for today, Professor Olivo Miotto, who gracefully agreed to give this talk in his busy schedule and huge time difference between the UK and Thailand. As I said, it's 12 p.m. in the UK and it's 7 p.m. at night in Thailand. So, Professor, thank you so much for taking time out. The title of the talk, as you can see on the slides, is Cryptotype: Discovering a Hidden Epidemiological Pattern in African Plasmodium falciparum, and you can see the nice African patterns on the screen as well. I can't introduce this man in a minute, but I'm going to try. I'm going to summarize the bio. I mean, obviously, Google him and you'll see his whole CV, but I'll try to do my best. Professor Olivo Miotto focuses on translating the massive quantities of data produced by sequencing thousands of genomes into meaningful knowledge about epidemiology of Plasmodium falciparum. Based in Bangkok, Professor Miotto runs GenRe-Mekong, a genetic surveillance project in collaboration with the National Malaria Control Programmes in the Greater Mekong Subregion. The project provides health bodies with actionable knowledge about drug-resistant strains to inform decision-making as well as interventions. Professor Olivo has a strong interest in the genetics of parasite populations in 4 continents, including Africa, and identifies patterns of evolution associated to responses to drug pressure and other human interventions. Professor Olivo collaborates with research groups across the globe to identify patterns in thousands of genomic sequences, seeking advances in our understanding of parasite evolution and helping us identify interventions that will win the struggle against the disease. Without further ado, Professor Olivo, thank you so much for joining us. You may begin your talk. Thanks, Professor.
Thank you very much, Pablo, and for that very flattering introduction. I was wondering how many of you I would know, but I can see a lot of new names. So for those who've never seen me or heard of me, I've been actually involved with Malaria Gen for a very long time. So you might be wondering, why is some guy from Bangkok working on African parasites? And the reason is because that's been true for the last 15 years. And as well as occupying myself with the problem of drug resistance in Southeast Asia, I've been more generally working together with the team at Sanger and with many collaborators all around the world on the global data set that is the Malaria Gen Community Project, both falciparum and vivax, although I generally tend to work a lot more on Plasmodium falciparum. And what we're going to be talking about today is some so rather surprising results that came out of analyzing the community project data. I've had some trouble sort of finding a name for this phenomenon. I eventually came onto the word cryptotype to indicate that what we are looking at here is not a strain of parasites that you can just recognize at a first glance. You need to do some sort of rather intricate analysis to find it. And hopefully you will agree with me that this is a very exciting new development. And I'll now proceed to tell you about it. If I can advance the slide. Yes. Okay, so what we're going to be talking about here is population structure. And I didn't really have a very clear idea as to whether you were all going to be population genetics experts or if you're going to know nothing about it. So I did put a couple of introductory slides. So don't be offended if this is too simple. Now, what is population structure? Why are we interested in it? So, generally, when we look at a population that is where mating is at random, then we expect all the individuals to be genetically somewhat as different from each other as each other. So they're all pretty much equally different, if you like. So, of course, if you have, your children may look more like you, but then the grandchildren look less like you, the great-grandchildren. And as we go to a large population, as long as people don't interbreed, then the population tends to look homogeneously different. What you see here is a neighbor joining tree. The neighbor joining tree, the length of the branches is proportional to the distance between the individuals, and the individuals are those little red squares. This neighbor joining tree is of nearly 400 samples from Congo. This is a very high transmission country where there's a lot of diversity of the parasites and there's a lot of random mating. So you can see they are actually equally spaced. Now, as you increase geographical distance, what you find is that there is some differentiation. So even if you take two countries which are really very close to each other, like Gabon and Congo, then you will see that parasites from the two countries tend to group separately. They're not, you don't see a sort of big mix of the two. You actually see that they branched off in this tree. And of course, as you increase the distance, the population structure increases. So this is a global tree that shows thousands of genomes from all over the world. And you can see that on the left-hand side, you have West and East Africa. And on the right-hand side, you've got the Greater Mekong Subregion, so the Asian parasites. And in the blue ones are from Cambodia, and the green ones are from Myanmar. And then you have places like New Guinea, Oceania there, and so on. So you get a lot of population structure when you look at large geographical distances. Now, the thing is, the geographical distance is not the only thing that gives us population structure. So there are other good reasons for looking at population structure in order to find non-random mating. And actually, these are the things that I'm more interested in. So I'll give you some examples of things that can generate population structure. One is geographical isolation, the well-known island models, where if individuals are on an island and they are unable to travel to the mainland, then they tend to inbreed, and so they give rise to subpopulations or even different species. This was actually Darwin's observation in the Galapagos Islands. But you don't actually need necessarily geographical isolation. Sometimes you have you have basically reproductive isolation. A case in point, for example, is when malaria parasites, falciparum or vivax, when they adapt to a new mosquito species. Since parasites reproduce sexually inside the mosquito guts, then a parasite that's adapted to a new species of mosquito can only mate with another parasite that has the same adaptation. As a result, they may separate in reproductive terms from the rest of the population. And you may get a population structure, but within the same geographical space, because they separate by the mosquitoes they're infecting. Another case is where you have low transmissions in countries like Cambodia, Vietnam, Laos, but now also in Africa, countries like Ethiopia, Mauritania, countries where the transmission is not as intense as what I showed you earlier in Congo. Then essentially people get bitten less, so it's less likely they'll be infected by multiple strains of the parasites. Therefore, there is less chance for strains to interbreed. So what happens in those cases is that mosquitoes pick up blood that contains multiple parasites, but basically these parasites are all clones. They are actually twins, but they can still mate with each other. So this is what we call selfing here, and it does produce population structure. And one particularly interesting case of population structure by selfing is where the parasites are under selection. A typical case it was in Southeast Asia with drug resistance, where in fact drug-resistant parasites, if they mate with other drug-resistant parasites, they tend to produce offspring that have a higher chance of survival, and this causes a population structure, particularly if transmission is low. So, in fact, in 2013, we published this paper which showed that even though when you look at population structure, even though most of Asian parasites, and you can see the label there where it says GMS, so those are Asian parasites, even though they cluster and separately from Africa, but in a sort of homogeneous group, there were clusters in Western Cambodia that separated out in this PCA plot that you see here. And basically, these clusters were clusters of artemisinin-resistant parasites. And even though at the time we didn't have the markers, we were able to establish that it was artemisinin resistance, drug resistance, that was causing this population structure. By the way, this is a PCA plot, and PCA plots basically are mathematical transformations that group together individuals that are more genetically similar to form a cluster. Now, we've used population structure in many different instances, and sometimes they can answer rather interesting questions. So, for example, a few years back, we investigated the emergence of some Kelch13 mutants, so probably artemisinin-resistant parasites in Papua New Guinea, and we use population structure in order to show that these mutants, which are circled there, the yellow little balls, they were not clustering together with the with parasites carrying the same variants, but from Southeast Asia, which are the blue parasites here. But also they were clearly they were not your typical Papua New Guinea parasites. They were very much in the middle between Papua New Guinea and Indonesia, Papua, Indonesia. So it did look like these parasites were the result of a mixture and probably some gene flow between the two countries. So these are of course the type of analysis that can produce information that then can be used for interventions by public health. And what about Africa? Do we have population structure in Africa? And the answer is yes, and I'm not the only one who's shown it. This is a figure from Alfred Mua's paper a few years ago, showing population structure in parts of Africa. And but what I was interested in, because again, population structure here is mostly by geographical distance. What I was interested in is whether we can see some of the other population structure in African parasites. So what I did was to take the MalariaGen PF7 data set. So this is about 20,000 genomes of Plasmodium falciparum, which have been sequenced with Illumina. And from these, I selected, well, the ones from Africa, of course, the ones that were higher quality. So in other words, they had good coverage across the genome, but also samples that were essentially clonal. In falciparum, you have a lot of mixed infections, where you have multiple genomes in the same sample. And whilst those can be very interesting in certain types of studies, in studies where you're looking for patterns, these tend to kind of blur the edges a bit. So we removed them in this case. This still left us with 4,376 samples, which is a decent sized data set. And they are genotyped by 700,000 SNPs. Once we filtered out the ones that were not present in Africa, the ones that were at very, very low frequency, and so on. So what did we find? So the first thing is I did some PCA plots, and very comfortingly, what I found was that when we looked at the first component, first principal component, which is where the most of the diversity is captured, yes, West Africa and East Africa were driving that component. You can see on the x-axis here, and Central Africa parasites were in the middle. So that was what was expected. But then you can see on the third component, which is the vertical axis here, then we saw clusters separate from the Gambia and Ethiopia, and unsurprisingly so, because these are countries that have very low transmission rates, and therefore these are the sort of countries where we would expect population structure to emerge. And in fact, in other components, the fourth component, the fifth, the sixth, and the seventh, we still saw groups of parasites from these two countries, and also from Sudan. These are the sort of brownish triangles there. We saw them separate from the main cluster of African parasites. So this was good evidence that there's population structure where there is low transmission, and that was good. But you might have noticed here that I left out the second component, right? And that's, of course, a very important component. But that's where we found the weirdness. So here, this is the plot of first component versus second component. And first component is West Africa, Central Africa, East Africa, like we showed before. But on the second component, we have this cluster of parasites separating out. And this was very different from the clusters in the other components because those clusters were from a single country. Whereas what we had here were parasites from multiple countries. Not only multiple countries, but some in West Africa, some in East Africa, some in Central Africa. So that's kind of a bit strange because usually when we look at population structure, it emerges because of inbreeding. But since some of these guys were from Madagascar and some were from Mali, it's a bit sort of hard to imagine that this would be interbreeding. So we looked at the distribution of these parasites, and you can see the countries in orange here are the ones where the parasites were sampled. And you can see that it goes from Mauritania, Mali, Gambia, all the way to Tanzania and Madagascar, all across the continent. And in nearly every country, they are at a relatively low frequency. They're something between 0.5 and 2%. So around 1 point something percent. And it's pretty much uniform all across. So across the continent, about one sample every 100 is similar to some in some noticeable way to to parasites that are probably about, in this case, 7,000 miles away. So what is special about these parasites? Are they different from parasites, the other parasites that are circulating in Africa? And the answer is they have differences. So here, what I've plotted is the FST. FST is basically a measure of differentiation. And here I've mapped it across 14 chromosomes of the falciparum genome. And on the y-axis for each chromosome, you have the FST value. So the higher the FST value, the more differentiated this cluster, AF1, as I called it, is from the rest of the African parasites. And here we're merging together West, East, and Central Africa all together. So you can see here that there are differences that are a bit everywhere in the genome, but actually there are regions such as this region on the right of chromosome 10, of chromosome 11. There are on chromosome 9, chromosome 2, for example. So there are many loci across the genome where this AF1 group is differentiated from other African populations. However, there are also large parts of the genome where, in fact, they're pretty much the same. I mean, look at most of chromosome 6, chromosome 3, chromosome 12, and over there you see relatively few differences. So it looks like these AF1 parasites are not clones, but they just have quite a lot of bits of the genomes that are identical amongst these individuals. And what are these bits? What are these SNPs which we found to be highly differentiated? Well, you can see here, actually, they are in sort of pretty well-known genes. There's genes like down here, there's MSP3, GLRP, SERA5, SERA6. These are very well-studied genes that, in fact, have to do with red blood cell invasion, but we'll come to that later on. So you can see these are very high FST. I mean, FST of 0.9 is pretty much like saying, and if you look at the plot here, that they're practically in all the AF1 parasites, and they are in none of the parasites in other parts of, not only of Africa, but of the world. You can see West Africa, Central Africa, East Africa, South Asia, West and East Southeast Asia, Oceania, and South America. So some of these mutations are shared by nearly all the individuals in this group, but they're completely absent or almost completely absent elsewhere in the world. In some cases, they might not be absent in the whole world. They might be common. For example, down here, the GLERP ones, you can see they're quite common in South America, but they are almost not seen at all in Africa. Okay, so essentially, these are very highly differentiated SNPs in many regions of the genome, and they are in only these sort of 1% of the parasites. And when we actually look at the correlation between these SNPs, it is actually very, very high. I mean, the correlation of 0.5 R squared equals 0.5, 0.6, 0.7. This basically means that if you know what the allele at one locus, you can predict the allele at the other one, pretty much. And you can see that these 6 loci that I'm showing here, which have pretty much 0 allele frequency anywhere but in the AF1 group, these ones are all on separate chromosomes, chromosome 2, 9, 10, 11, 13, and 14. So we have different parts of the genome physically separated, and they're basically presenting these unusual alleles all together correlated. So just to put in the context how remarkable that is, you know, I should like to remind you what happens when you recombine. So, you know, in falciparum, you have sexual mating. So when there is a recombination of one of these AF1 parasite, which is represented here by a green bar, then the offspring will have only about half of that genome, and half it will get from another one. And then at the next round of sexual mating, you will have much less, and so on. As time goes by, you end up with only just little tiny bits of the genome. And think about it. If this let's call it strain, for lack of a better word, if these AF1 parasites are distributed all over Africa, they obviously haven't emerged last year. They've been around a very, very long time. And if they've been around a very long time, you'd expect the haplotypes to be reduced to almost nothing. And instead, here you have many regions of the genomes that are coming together. So one thing that we asked then is yes, these bits of the genomes are coming together, but are they emerging independently, or do they come from the same ancestry? By the way, I don't mean that they all come from the same ancestor. What I mean is that all AF1 parasites for any one locus are inheriting from the same ancestor or not. So we use identity by descent analysis, a program called HMM-IBD. Many of you will be familiar with this, but for those who aren't, what HMM-IBD does is it takes 2 individuals, it looks at their genomes, and it looks for stretches of the genomes that are unlikely to be, that are so identical that it's unlikely that it's happened by coincidence. And so it predicts that that region of the genome has been inherited from a common ancestor. And so I did some identity by descent analysis of the AF1 parasites, and I looked at the proportion of the pairs of parasites in AF1 that actually shared a common ancestry. And here is a plot again of the 14 chromosomes, where you can see I've highlighted in pink those regions where more than 50% of the pairs of parasites in AF1 are in IBD with each other. So they're likely to have inherited from the same ancestor. And you can see some of these regions are massive. If you look at the ones on chromosome 10, but also on chromosome 9, and the one in the middle, chromosome 6, and those on chromosome 14, they are actually very large regions where, in essence, all the AF1 parasites, to first approximation, have inherited that stretch of the genome from the same ancestor. Okay? And if you remember the previous map of the differentiated alleles, you will remember that they pretty much fall in the same regions as this IBD. And when we looked at this, there were about 23 regions of high IBD, so where more than half of the parasites are inheriting from the same ancestor. And I've kind of ranked them by how differentiated there are. So we have now 23 regions that appear to be inherited by this AF1 parasite. Again, this is totally remarkable, because if you think of recombining with a wild-type parasite, then half of your genome evaporates, so the chance of inheriting all 23 is minimal. And so actually the chance of passing it down so it can be maintained in the population is really quite an amazing numbers game. Now, you might say, well, I mean, actually it's not that difficult. All that happens is that the AF1 parasites only mate with AF1 parasites. It's all inbred. But actually, that's not so. And so first of all, this panel A shows you that the level of IBD that's seen inside the AF1 group is massively larger than the average in Africa. Most pairs of parasites in Africa, when compared to each other, have almost no IBD, because basically recombination breaks down the identity very rapidly because of high transmission. So basically, you can see that in West Africa, Central Africa, and East Africa, WAF, CAF, and EF down there, you have a median of IBD, which is near zero, whereas it's over 20% inside the AF1. However, when you look at the next plot, what you can see is that the group AF1W, so the West African AF1, has more IBD with the West African parasites than it has with the East African parasites. And the vice versa, the opposite is true for East Africans. So essentially, we are seeing the evidence of recombination with local parasites that are not AF1, okay? But it evidently doesn't happen very frequently, and the AF1 parasites are evidently able to maintain their compendium of variants. And when we use IBD proportions to as a distance measure in order to do a PCA, you can see that our AF1 parasites shoot off from the main group of African parasites. And you can actually see here that the East is somewhat different from the West in this group, which is what we were showing earlier on. And you can also show the same when you use a neighbor joining tree. So this is a summary, and this is the type of things that you like to put in the genomics paper. You can see the circle here represents the 14 chromosomes, and the blue lines actually show the R-squared, the correlation between alleles, and the little red boxes inside show the high IBD regions, and then the green band here with the orange dots represent the highly differentiated variants. And as you can see, it all comes together. Basically, the regions that are in highly correlated with each other are also in high IBD, and they're also highly differentiated with respect to the rest of the African parasites. So when we look at the 23 regions of high IBD, that's what the columns are here. Do all the AF1 parasites have the same variants? Well, almost. You can see here these are the 47 AF1 parasites that I found, and where they show an orange box, it means it's the AF1 variant. A blue box is the wild type variant. And what you can see is that if you take the first few regions, if you take the first 7 regions, practically all of them do not have a wild type in that region. And then the sort of purest forms of AF1s share about 20, almost 20 different regions with the same variants. And those come from Ghana, from Madagascar, from Uganda, from Congo, Mali, Ivory Coast, etc. So yes, there is evidence of recombination. Yes, there is evidence that some of these AF1 parasites do not have a complete set, but generally they tend to be quite amazingly similar. And what is in these 23 regions? So these are the top hits. And as you can see, they're actually pretty large regions, some of them. Some are small, some are larger. The top one is on chromosome 10. And what I like to highlight is in some of these top ones, a number of the genes that are present. So those boxes are the genes, and together with their names. And you can see that a lot of these genes are colored in red. And genes colored in red have to do with the invasion of red blood cells. And the ones covered in brown, which is another big lot here, have to do with getting out of red blood cells. So invasion and egress. And then the third biggest group is are antigens that are expressed on the surface of the red blood cell. In other words, when we look at the top hits of this compendium of variants, they're all in genes that have to do with interacting with the membrane of the erythrocyte, either going in or coming out or making things stick out of it, basically. So what does that tell us? Well, it probably tells us that this has something to do with red blood cells. There's some sort of specialization that these parasites have. But let's first of all look at some of the amazing features of the biggest region there. And what we found, this is a coverage analysis that compares AF1 parasites with West and Central and East African ones. And you can see that there is basically a big deletion in chromosome 10, which has 2 whole genes, MSP6 and another MSP gene. And what looks like a piece of this gene called DBL-MSP. And when you look a bit more closely, yeah, you can see here that DBL-MSP seems to kind of being cut in half. But then you can have another clue when you look at the end of, on the right-hand side here, you have another gene called DBL-MSP2, and this is a clue. There are two paralogs. And this one seems to have a first half that's highly covered, more highly covered than the downstream half. And to cut a long story short, what's actually happened is that the two genes, DBL-MSP and DBL-MSP2, have basically recombined, forming a new version of this DBL-MSP gene. By the way, DBL-MSP and DBL-MSP2 are well known to recombine even at this site, but I believe this particular variant is one that has not been described before. And you can see down in panel B here, you can see that this recombination happens because the two genes have a completely identical stretch of the gene, which is of the sequence, which is shown in green there, and therefore they've sort of crossed over and recombined. And on chromosome 9, similarly, we have a sort of very large haplotype, which has got to do with the MSP1 gene, which is the gene that makes the first contact with the red blood cell. And what we have here is a haplotype which is very uncommon in Africa, but is not so uncommon in Southeast Asia. So it might have originated over there. However, even with respect to the one in Asia, there are some differences that are not seen anywhere else. So jury is still out as to what's going on there. And what I just wanted to stress here is that not only are these genes all involved in invading or exiting from red blood cells and doing stuff with it, but they are actually interacting with each other. They're stacking up with each other in order to create structures that basically attach the merozoites to the erythrocytes. So what we may be seeing are sort of variants of these genes that allow them to stick together and to form new forms of these structures. So I'm practically finished here. I just wanted to tell you my hypothesis here, and basically it is that these parasites are specialized in some ways. They're specialized probably to fit in with some human blood phenotype. Some specialized to invade a particular blood group, or perhaps a hemoglobinopathy that is protective of against malaria. But they have incredible characteristics. They're all over the continent, which means they've been around a long time. They're very functionally specific. They are very low frequency, so if they do occupy a human niche, this niche is quite small, but very advantageous, because otherwise they would have been removed from the gene pool. And in future, what I'd like to do is work with collaborators in Africa. We got lots of friends down there, and try and find some of these parasites, put them in culture, test them, do some invasion assays, but also work on some new bioinformatics method for discovering cryptotypes. It could well be that this is not the only cryptotype there is. In fact, this one was relatively easy to find because it's so highly differentiated. But this is not a requirement for a cryptotype. It could be cryptotypes that are not completely absent from the main population. Okay, and with that, I would like to thank all the partners. These are mostly the authors on the paper that's gone to preprint. Now, none of this could have been done without the Malaria Gen whole genome data set. It's an immensely useful, totally important data set. And I like to remember that it was Dominik Kwiatkowski who actually launched the whole thing. So thanks to him and thanks to all the collaborators. Thank you very much.
Hey. Thank you so much, Professor Olivo, for a brilliant presentation, very informative and quite accessible. We now have time, about 13 minutes for Q&A. So please do type your questions in the Q&A box or in the chat box, and I'll read them for Professor Olivo and he'll answer. So I'll read the first question, Professor. It's from Kuchu Boris Tamege saying, hello, Prof. I'm Boris Tamege attending from Milan, Italy. The question is, what will happen if the ineffective Plasmodium falciparum mates with the non-malaria mosquito here in Europe? Professor Oliver?
Okay, well, I'm not sure how many Anopheles we have in Europe right now. So, sorry, what is the question is specific about Europe?
I think it's about Europe, yeah. I can reread the question. What will happen if the ineffective P. falciparum mates with non-malaria mosquito here in Europe?
I'm not entirely sure how to parse that, but the likelihood of that happening is very, very low because we just don't have those vectors in Europe, or at least not to a significant level anyway, not anymore.
Thank you so much for that answer, Professor. Another question, or rather two questions from Madi Safapur. Thanks for the great talk. How alleles on different chromosomes can be highly correlated if we assume that during the meiosis, the chromosomes are independently transferred to new gametes? And my next question is about the kind of insights such as high correlation can provide us. Can we use this information to stratify our population into different subpopulations? Oliver?
Yeah, and I mean, to some extent, this is what I've done here. Now, I mean, the first part of the question, I think it's $1 million questions. How can you explain that? Not just, I mean, it'd be hard enough for two loci to be correlated in spite of recombination, so that they're not lost along the way. But 23. That's a massive number, and you can probably work out some probabilities there. But I think there's a variety of possible answers. So if you ask, I don't know if Mara is with us here, but one thing that she has suggested is that maybe it's not 23, maybe it's fewer that have rearranged together, and genomic arrangements, they are actually easier to inherit together because they're now located closer to each other. But I think that's probably not even necessary. What is happening with these parasites is I think they're playing the numbers game. They are mating, and from time to time, very, very low probability, they manage to get some offspring with the whole compendium, and these have some enormous advantage which we don't know what it is, but that enables them to still proliferate at a certain level. That's my thinking on this, but it's not an answered question.
Thank you so much, Olivo. I see there's a question from Jody Lendia. I see your hand is also raised. Jody, do you want to go ahead and ask your question? Do you want to unmute and ask? Jody, go ahead.
Jordi?
Jody, we can't hear you.
Okay.
Otherwise, I'll read your question quickly, because I think I saw you asked it twice. So this is from Jody Lendia. Hello, Oliver. Thanks for a great talk and exciting findings. Can you date the period of existence of the potential common ancestor for AF1? If I remember well, the origin of Plasmodium falciparum is thought to be from gorilla Plasmodium roughly 10,000 years ago. Is this compatible with your findings? Professor Oliver?
Yeah, I think it will take some better geneticists than me to try to compute that. We haven't tried it. Of course, it'd be an interesting thing to do. I don't know what the answer is, and it's anybody's guess whether that predates gorillas or not. Now, one interesting thing is that some of these haplotypes that we're seeing here are also seen in South America. And so one possibility is that this is actually at least older than the migration that brought falciparum to South America, which would have been at least sort of 500 years or something.
Okay, thank you so much, Olivo. There's a question from Jacob Amalgo Garcia. Great talk, Olivo. A speculative question here. What do you think would be the fate of AF1 and other cryptotypes if transmission starts to go down in Africa? Do you think they will quickly vanish from the general population, or would they become easier to spot?
I honestly have no clue. I suspect they would struggle to survive because basically they would have a lot less chance of playing the numbers game. But yeah, that's something that some modelers could try answering, I think.
Thanks, Oliver. Another question is from Irene Omedo. She's asking, did you conduct any temporal analysis to see whether the frequency of this parasite subpopulation is increasing over time?
Yeah, that's just simply not been possible because the numbers are so low. You know, you have to sift through 4,000 parasites to even just see them. You know, so it's not it's not really a problem that can be tackled, I think, at the moment.
Okay, thank you so much, Professor Oliver. We have about 2 more questions that I'm seeing in the chat, but before I move on to them, I'd like to welcome my colleague who also works closely with Oliver, Christina. I don't know if you have any questions or comments from your end before I move on to the other questions.
Thanks, Pablo. Oliver, thank you so much for your presentation. It's been excellent and is such a fascinating theme. And I think others will agree with me that if Dominic was to see this, he would be thoroughly excited, and he would be with his eyes just like completely blown away by all of that. My question is, do you think, can you hypothesize what would be the origin, the original, from which country that those F1s could have been? If it's at least East Africa, West Africa, or can you hypothesize?
No, I did ask myself that question, but I think, I mean, I'm not easily seeing any bias. At first, I for a long time I thought they were originally from West Africa, but then I realized that all the evidence for that was simply to do with the fact that we have so many more samples in West Africa, so we had stronger signal. I cannot honestly say where I think they come from.
Thank you.
Thank you so much, Christina. So we have about 5 minutes left, and I'm going to ask that if you have a pressing question, please do type it quickly in the Q&A box.
Antoine is asking something.
Yes, hi, Olivo. Thank you so much for this talk. It's a node to hypothesis-free science, I think. My question is about, so far you've only analyzed the monoclonal infections. Have you looked for AF1 in polyclonal infections?
Yes, I mean, in the paper, I've only included monoclonal because that's just easier to handle. But actually, when I started this off, I did start with the whole data set, and there's plenty of polyclonal ones, or rather, there's plenty where AF1 parasites are together with wild-type parasites. So, and pretty much as you would expect, given their frequency.
Okay, thanks.
Thanks, Prof. A question from Ming Tang. Thanks, Prof. Olivo, for your insightful presentation. Just curious, how would you describe the differences in the Plasmodium falciparum resistance landscape in Africa versus general Mekong subregion or Southeast Asia region, and your thoughts on the strategies required for genomic surveillance to mitigate the drug resistance problem?
So this is a completely different presentation altogether, I think. I mean, this is about drug resistance. Yeah, I mean, I don't think we have a lot of time to answer this, but I mean, Africa is such a diverse landscape for malaria, where you have regions that are quite similar in epidemiological terms to Southeast Asia, and there are others where, you know, there's a completely different situation where the same approach cannot be applied. So it's a very long discussion, this.
Okay. Last question that I'm going to ask you, because I'm looking at the time and I see more questions are coming in. So apologies to those of you that are not going to get a chance to ask your question. I'm going to ask a question from Dr. Victoria Simpson, who's saying, Dr. Olivo, what advice do you have for the next generation of parasite genomic data scientists, specifically people who are newer to the field? Professor Olivo?
What? Sorry, I missed some of that. What?
So the question is from Vicky. What advice do you have for the next generation of parasite genomics data scientists, specifically people who are newer to the field?
Come and visit me in Bangkok and spend time here. If anything, I have difficulties finding people who, who want to get in deep onto this. Well, I mean, personally, I think we are sitting on a huge treasure trove of data, and I think if this study proves anything, is that you can just take it, slice it any which way, and find amazing things. So now, this is not, I mean, my first advice is take the data and play with it. Now, one problem is it's quite difficult to play with, and this is where probably, it would be of benefit to come and visit me in Bangkok. It's a nice place as well. But yeah, we probably need to do better to pass on the knowledge of how to data mine these data sets. And there's probably hundreds of ways that I'm not exploring. There's probably, you can probably use all sorts of artificial intelligence and data mining approaches and so on, which I don't know how to do. But yes, I think we should do more to make the knowledge available to young people who want to do this.
Thank you so much, Professor Olivo. I'm looking at the time, and apologies to those of you that didn't get your questions answered, but you can totally get in touch with Professor Olivo on the side and engage him more on the topic. Professor Olivo Miotto, thank you so much for your time. I know it's very late in Bangkok, and I'm sure the weather is amazing compared to the UK, but yeah, we do appreciate your time and effort. I know you're very busy. This was a brilliant and informative presentation. Thank you so much, Professor.
Well, it's been a real pleasure, and I'd like to thank you for inviting me. It's been great.
Thank you so much.
Thank you, Olivo.
Thank you.
Okay, everyone, have a good evening, and we'll see you next month for a different journal club. We'll send the invitations in due time. Have a good day, everybody. Goodbye.
Bye. Thank you.